Okay, let's talk about von Willebrand disease. I will abbreviate it as VWD. So the main problem in this disease is there is problem associated with VWF. So von Willebrand disease is problems with von Willebrand factor. So F over here is for factor. What is VWF? I will talk about it sooner. So quickly, let's see what VWD is. So primarily it is autosomal dominant, but it can also be autosomal recessive. Okay, then there's also one more version of it where it is acquired. Okay, so we can have three different versions of VWD. When it is acquired, it is also called as AVWS, acquired von Willebrand syndrome. Okay, there are multiple causes of it, but we'll focus more on the autosomal dominant one. So von Willebrand disease is a bleeding disorder. It's a bleeding disorder. So when I talk about bleeding disorder, there can be different ways how bleeding disorder can present. Von Willebrand disease, however, is a type of thrombasthenia. Okay, thrombasthenia simply means, so thromb is for the platelets, right? So platelets function is decreased in this case. Then there's also term, it is thrombocytopenia, where the number of platelets are decreased. So in this case, it is a thrombasthenia, right? It's an important one. Okay, now what actually it is? So in this condition, because it's a bleeding disorder, so the person will have bleeding, right? And most of the time the patient have asymptomatic condition, or he can present with mild form, or most of the time it is unnoticed. Okay, until unless the patient have some sort of bleeding, mostly in the bleeding prone areas, most of the time when the person eats. Okay, and because of that, if there is some minor trauma to it, there can be bleeding. Okay, and because of this the patient may have bleeding disorder, bleeding, right? So the patient may present in the very early scenario with epistaxis. These are just minor symptoms. Okay, then there can be menorrhagia, uncontrollable, I would say, not so much severe. Severity depends upon the type of VWD. Then there can be mucous membrane bleed. Mucous membrane bleeds. Okay, so these are the three most common. Menorrhagia, again, much more common in females, not in males. Good. Now, we just said that there was a problem with VWF, right? So first of all, let's discuss what actually is VWF. Okay, so let's discuss about VWF, and then we'll talk about different types of VWD and what's the problem associated with each of them. So VWF is a type of factor. So von Willebrand factor. In this disorder of VWD, either the function is lost, either the production is decreased, and so and so. So it's a factor which is expressed in different areas, okay, of the body, and it helps in overall platelet aggregation and adhesion, it is collection and sticking of platelets. So platelet aggregation and adhesion. It also helps in stabilization of factor 8. So factor 8 stabilization and protection also. Then it helps to bind to subendothelial collagen and GP2B3A of platelets. Okay, so overall it takes an important role in hemostasis, right? So all these three functions takes major important part in hemostasis. So very important. Okay, star. So VWF, the decrease or overall function decreases, they somehow target the hemostasis of the patient. So let's see how they are formed. So VWF is distributed in different parts of the body. It is majorly found in three different ways. Okay, it's distribution. So distribution of VWF is in three different areas: endothelial cells, then the platelet itself, and in the plasma. Three forms. Okay, now okay, so in the endothelial cells, they are formed within a vesicle. This is a vesicle which is called as Weibel Pallade bodies. Okay, I'll discuss and I'll just show you the picture of how it actually looks like. Then it is associated with the platelets, and for the plasma it is circulated in the free form. So free form circulation where it binds to factor 8. Okay, and it protects, right? So this thing binds to factor 8 and stabilize it and protect it. Okay, so let's see how actually it functions. So let's say we have a platelet, not like this. Okay, one platelet, second platelet, and it has a GP2B3A. This is a GP2B3A expressed on the platelet. So this VWF factor, okay, this is a VWF factor. VWF factor. It allows them to stick together and aggregate and adhere to each other. So platelets. So this is the first way. Second, it stabilizes the factor. Okay, so let's say this is your factor 8. Without it, it destabilizes so fast. So von Willebrand factor. So this is a von Willebrand factor, okay, that binds to it and stabilizes it. Factor 8 and stabilization. Hope you guys are familiar with the coagulation cascade. If not, I will sooner, hope so. I will make a video of that as well. And lastly, it is expressed on the endothelial cell. So endothelial cell, how it looks like. So this is a blood vessel, and within the blood vessel, the innermost lining, the innermost layer, is the endothelial layer. Okay, let's take this cell. Okay, so this is an endothelial cell. Within this endothelial cell, there is a vesicle. This is a vesicle called as Weibel Pallade bodies. So it produces glue for two different things. Okay, it is somewhat sticky material, so sticky material in two different format. Either it is a VWF or it can be a p-selectin. We'll focus more on the VWF right now. So this VWF will be within the variable polarity bodies. Once there is a damage, okay, let's say here a damage occur at this level, so there will be damage to the endothelial cell, and because of this, the layer that is at the lowest level, which is called as subendothelial layer, that is sub means below, endothelial layer, right? So let's say this is a subendothelial level, and the blue color over here is the collagen, subendothelial collagen. This is subendothelial collagen, and toward this side is the lumen, okay, of the blood vessel. So once this happens, the vesicle fuses to the outer membrane, okay, and these red dots, which are the VWF, will be expressed onto these surfaces, okay, so that platelets, these platelets again can come over here and attach themselves onto the wall of endothelial cell, damaged endothelial cell. And also the same VWF will help other platelets to aggregate and adhere to each other, and overall a clot forms, okay. So this is how the VWF take an important role in all of it. Okay, now one more thing before moving further, that the form of presentation of VWF. So VWF form is a polymer type of multimer. It's a multimer. What multimer is? Multimer is something which is made up of many other monomers. So here it is, it's a multimer, not tetramer, not, it's a type of polymer again. So even bigger, so many clumps together. So this is VWF multimer. One single circle, which I donated over here, this one, okay, let's say this one, okay. So this one single unit will be a VWF monomer, right? So naturally in the body it's a multimer, right? So this format, it's found in the body. Now, so the VWF factor, it differ in size within the body, present in human body, right? It can be big, it can be small, but it will be a multimer again, okay? So and the size will again depends on different other factors. We'll not discuss all of that, okay? So because we just said the size of VWF differ, right? So it can be big, it can be small. Let's make it. So this one is a small multimer, small VWF multimer, okay. And as we go on to the spectrum, the size will keep on increasing, okay. So this is the most bulkier VWF factor present in our body, okay. These are all just representations. I don't even know how it actually looks like, okay. For that we have to go in the molecular biology. Anyways, this is the bulkier or the largest or the biggest size of von Willebrand factor, okay. and within the spectrum the size will keep on increasing. There will be again this level which is the intermediate level. Okay, so this is the intermediate level. It will keep on increasing in size. It will keep on increasing. Okay, so this will be much more bigger. Anyways, you got it right. So let's make one more. Anyways, so this level will be the intermediate level. Okay, so one basic theory: the size of multimer, more will be the size, more the number of bonds it can make with platelets. So the number of bonds formed with platelets. Okay, more bigger the size, more number of bond. Lesser the size, less number of bond and less platelet can adhere. Okay, so normally we have this biggest and the intermediate level, which are mostly functional in the hemostasis. So the largest among them takes part in the, not the largest, the intermediate to the largest, they take part in the platelet adhesion and aggregation, much more nicely aggregation. Okay, so we are done with this one. Now let's discuss about the disease. What is actually going on with the patient right now? So so for the VWF, it is of 3 different types. Again, major 3 different types, but there are about hundreds of varieties. Okay, well, let's write it somewhere. VWF, von Willebrand factor itself, okay, is 100 different types are present. Out of them, okay, out of all those 100, only few have the potential to cause a disease, out of which we only need to remember 3 different types. Okay, so type 1, type 2 and type 3. Okay, so in the very earliest slide I mentioned that it can be autosomal dominant, it can be autosomal recessive, and for the acquired one, when patient acquires it later in the life, it is mostly because of autoimmunity, that is antibodies against VWF. Okay, and this causes the destruction or at least decrease in the number of VWF, and the same presentations happen. So when the platelet won't able to adhere with itself and its nearby platelets, overall the cord will not form, right? And because of it, the patient will have bleeding, right? Because the platelets are one of the most important things takes part in hemostasis, right? So we were talking about VWF. There are 100 different types. 3 of important are the type 1, type 2 and type 3. So most common among all of them, type 1, about 70% of the cases. Then second most is the 25% for the type 2, and the type 3 is again rare, right? Being rare, it is more severe forms of VWD, right? And the disease severity for the type 1 is, it's a mild form. Okay, mild form of disease, and it is a severe form of disease. For the type 2 again, it is a mild to moderate. Okay, now in the very first slide I just talked about it can be autosomal dominant, recessive and mixed. Remember, So the type 1 was autosomal dominant type. Type 2 also, it's an autosomal dominant type, and for the type 3 it is autosomal recessive type. Okay, so what is autosomal dominant, what is autosomal recessive? I have the videos already uploaded, watch it. Okay, same presentation, male to female ratio again 1:1:1, and so and so. Now, important point, the type 1 is a quantitative variant of VWD. So okay, so the quantity form, quantity of VWF, this decreases. Okay, for the type 3 there is almost very low to about none level of VWF, and because of this, the severity, right? So severity depends upon how the VWF actually is, whether the amount is decreased or the function is decreased. So this is a quantitative variant. Same for the type 3 variant. So quantitative, this is also quantitative variant. Okay, same type 1, type 3. For the type 2 specifically, the amount, or let's not write amount, let's say the quantity of VWF is normal. So if it is normal, so why it's a disease? Overall function is decreased, so there is a decreased activity, that is the VWF form, okay, is not functioning properly. And why it's not functioning properly? Because the multimer formed, which are basically protein, they are defective. Multimer formed are defective. One more thing I just forgot to mention, that von Willebrand factor, right? Von Willebrand factor, let's write it over here. Von Willebrand disease is a disease of von Willebrand factor, and von Willebrand factor is a type of protein, right? So it's a protein, it will come from some gene, and its gene is located on chromosome number 12. Which gene? Very easy, von Willebrand gene. Okay, so yeah, if you just want to remember, you can remember this, right? Okay, so why it was like this? Because of, in all these three scenarios, because it's a genetic condition, in all three of them, either it can be AD or AR. In both the condition, again, chromosome number 12 is affected somehow with the affected von Willebrand gene. Okay, von Willebrand gene. Okay. Now, among the type 2, okay, there are again different types of type 2 with that we don't need to remember. At least you can remember, let's write it over here. For the type 2 also, it is 2A, 2B, 2M and 2N. Don't worry about all A, B, M and so and so, just remember this one. This is the most important one, type 2A. So let's write it over here. Type 2A is the most critical and disastrous form of type 2. And among all the different variants of VWD, uh, oh sorry, I just did something bad. It's not VWF, it's VWD. So VWDs are three different types. Okay. So the most severe among all of it is the type 3 with the very low to about none levels of VWF. So the severity depends upon how much the VWF actually exists and how functional they are. Right. So yeah, we are done with this one. So now let's talk about its clinical feature and how to diagnose it. So clinical aspect or the clinical features of it. So primarily bleeding disorders, so there will be bleed, mucocutaneous bleed, most commonly within the mouth, oral submucosa, mucosa. Then the same for the nose, epistaxis, mostly in the dried climate or the picking of the nose can cause it. Then in the woman, during the menstrual cycle, uncontrollable flow, menorrhagia, right? Then GI bleed can increase. Then post-operative bleed, mostly people don't come to you saying that I have this disease. Mostly they come to you because they're like, doctor, my bleeding doesn't stop. Okay, it just keep on flowing. The clotting time actually is increased. So post-operative, once the surgeon when the surgeon operates on you and they find that your bleeding is not stopping in the time it should, so post-operative bleed. So yeah, this is the clinical feature. In short, any condition which can cause minor trauma and you have bleeding, it can be one of the reasons. And when I say one of the reasons, so let's talk about the differential diagnosis. So it's a bleeding disorder. So whenever patient comes to you, you don't simply just say that, oh, you have one Willebrand disease. There can be multiple other different causes of it. So first cause, hemophilia A, hemophilia B, hemophilia A. Hope so. It is. Hope you remember that it is factor 8 deficiency, right? For the factor B, it is factor 9 deficiency. Now, overall diseases which causes the platelet function disorder, that is the thrombocythemias, among which this is one of the conditions. So, platelet function decreases. It can be due to Bernard-Soulen syndrome, okay, which is a defect in GP, not 2B, 1B, 9 or 5 complex. We'll talk about this disease someday or other. Next is Glanzmann thromboasthenia. So platelet function decreases itself is a thromboasthenia. Okay, and this is a defect of GP2B3A, Then what else can be the differential? Let's say the body is not able to produce thrombocytes, so it can be due to liver disease, cirrhosis, okay, liver disease. The factors that causes the maturation and the formation, maybe they are decreased, so overall liver diseases, okay. Then it can be an acquired version of it, acquired von Willebrand syndrome, majorly in lupus, okay. So there are some conditions in which antibodies are formed, right? So we can check for those antibodies. Then there can be overall vitamin K deficiency. Right, and then there can be overall thrombocytopenia, right? So liver disease in which the factors are not produced, even the factor which are responsible for the production of thrombocyte itself, which can cause thrombocytopenia. Okay, and also factor 13 deficiency. Okay, now being said that about the vitamin K deficiency, right? Vitamin K and factor 8. So one point that you should know that von Willebrand disease is a disease of intrinsic pathway. Intrinsic pathway. Why intrinsic pathway? Because in the intrinsic pathway there is factor 8. So factor 8 and von Willebrand disease goes hand in hand. Okay, so we remember that von Willebrand disease was a disease of von Willebrand factor, and this was stabilizing factor 8, and in the absence of von Willebrand factor, or at least the decreased levels of von Willebrand factor, will causes the decreased level of factor 8, and again it will causes the symptoms by delaying wound, delaying delayed clotting time or delayed bleeding times, which means the patient will bleed more, and if the patient will bleed more, it's a bleeding disorder. When the patient have some bleeding disorder, we as a doctor, we need to find in which pathway, in what structures are involved that is causing the patient problems, right? Okay, now how to make the diagnosis. How to make diagnosis? First of all, because it's a genetic problem, majorly. Okay, I accept it is acquired, but it is again not that much common as it is a genetic one. So for that, again, family history must be positive. People within the family will have the same symptoms, okay, irrespective of being a male or female. Then signs and symptoms will be again positive for the patient. Then we can order some lab tests in which we can check for the CBCs. Okay, all the different types of cells can be normal. Okay, because it's a von Willebrand disease, even the thrombocytes level will be normal. So we can check to rule out at least it's not a thrombocytopenia, because we discussed in the differential diagnosis that thrombocytopenia can be the one case which can cause the bleeding disorder, but we over here we need to rule out the von Willebrand disease. So we can go with the CBCs, then we can check for the INR. Okay, so the PT will be normal in this case. Now because von Willebrand factors was stabilizing factor 8, which is responsible for intrinsic pathway, right? So PTT, which is responsible to find whether there's a problem with intrinsic factor, and the PT is for extrinsic pathway. So because there was no problem in the extrinsic pathway, again it is normal. Okay, for the PTT, because there is a problem with factor 8 now, so PTT will be prolonged for this disease. Okay, then we can directly check for the VWF antigen itself. Okay, how much actually is it? So quantity you can check. Then we can check for the overall activity. Okay, so that we can rule out the type 2 variant of it. There is a specific test for the VWF activity, that is the VWF is to RCO. Okay, what is this RCO? Ristocetin cofactor activity. Okay, just remember the name, no need to go so much in depth for this. Okay, then we can check for the overall factor 8 availability and its activity. So factor 8 availability and its activity, but not so much important, but again we can. And lastly, we can check for the multimer analysis, multimer of von Willebrand factor. So we can do all these things, right, to check whether the patient has this disease or not. Now, let's say the patient has this disease, and now we just finally concluded that, yeah, now you are diagnosed with von Willebrand disease. Now, how to treat it? First of all, avoidance. First thing, because you cannot help, you cannot cure. Okay, we can only manage. Okay, now how we manage it? Okay, so for the management, there is one magic pill which can be given, that is the desmopressin, abbreviated as DDAVP in many areas. Now what the hell is this DDAVP, which is a chemical name? Don't need to remember, but you have this thing inside you that, no, I have to know everything. Then yeah, it is one, let me check it, D-amino 8-D-arginine vasopressin. So DDAVP. No need to remember, just remember this one. This is a magic pill. How? Because yes, it's a variant of ADH, antidiuretic hormone. Which type of variant? It is a synthetic analog. Synthetic analog of antidiuretic hormone in the body, desmopressin. Remember vasopressin? So desmopressin is one of it. So this actually releases, yes, it take part in the antidiuretic hormone activity also, but also one more function. It releases the stored von Willebrand factor from endothelial cell. Okay, so yeah, we can manage somehow. Okay, this can manage somehow, but if there is almost no production, it is in the type 3 variant, it can be a little bit problematic. So in that condition, what we do, we give factor 8. So factors, or we can simply write pre-formed factors separated from plasma can be given to the patient. Okay, so yeah, this was about the VWF and the VWD. Okay, so thank you for watching, and I will try to start with the skeletal diseases of the autosomal dominant disorders. Yeah, okay, thank you for watching. See you in the next video.